Hello everyone, myself Vaishnav Mane from TYPSC. In this video, we'll talk about generalized transduction. So let's start by learning what is transduction. Transduction is a process by which bacteriophages transfer genes from one bacterium to another. Such phages are called as phage vectors. Means the phage vectors are like transporters which carry genes from one bacterium to another. So the genetic material that is transferred during transduction process is less than 1% of the total bacterial chromosome. So the recombinant recipients are called as transformers. That is the bacteria which take up the genetic material from the phage vectors are called as transformers. As we've already known, the lytic and lysogenic cycles of the bacteriophages. So, in transduction, the lytic cycle of the bacteriophage plays an important role. So, let's see about lytic cycle of the bacteriophage. When the phage infects the host cell, it introduces its DNA, that is the phage DNA, into the host cell. This leads to the digestion of the host DNA. Thus, the host DNA is broken down into fragments. So, then the phage DNA replicates. The host DNA transcribes and translates into phage DNA, producing phage proteins. So, here the phage proteins are the body of the bacteriophages. Then, these phage proteins assemble and form bacteriophages, and then the lysis of the cell takes place. Thus, the new phages that are formed are released outside. Thus, the cycle continues as these new released phages infect other host cells. There are two types of transduction. First is the generalized transduction, and second is the specialized transduction. In generalized transduction, any genes can be transferred from one bacterium to another. But in specialized transduction, only specific genes are transferred. Specific genes in the sense the genes which we want, or the genes which have specificity towards the phage protein. So generalized transduction. The first generalized transduction was discovered by Joshua Lederberg and Norton Zimber in 1952. So, when they were trying to produce recombinant strains of Salmonella typhimurium, they did an experiment which used the Davis U tube. So, for what they did was they took two strains of S. typhimurium, which were auxotropic to certain amino acids which are essential for their growth. So, one strain was auxotropic to methionine and histidine amino acids, while the other strain was auxotropic to phenylalanine, tryptophan, and tyrosine amino acids. So, he did this experiment using two different methods. So, let's first see them. Here, first in the first method, these two strains of Salmonella typhimurium, which were auxotropic to methylene, histidine, phenylalanine, tryptophan, tyrosine, respectively, they took these two strains and mixed them. So they incubated this for a certain period of time, and then plated this mixture onto the minimal medium. So the minimal medium didn't contain any of these amino acids. Like these amino acids were absent in the minimal medium. But they still got the prototropic colonies, that the prototropic colonies could produce all the five amino acids which were needed for the growth, and thus they grew. So, why did this happen? So, the conjugation takes place when two bacteria come in contact with each other. So, this was due to the conjugation taking place. As the conjugation took place, the genes coding for these two different amino acids, which were auxotropic, were exchanged between the bacteria, and thus we found the prototropic colonies which could produce all the five amino acids. This was the first method which did it. In the second method, similarly, they took these two strains which were oxytropic to some amino acids, and they put these tubes, put these strains in the Davis U tube. That is one strain on one side of the Davis U tube and the other strain on the other side. So, and this Davis U2 had a filter in between. So, this filter did not allow the any bacteria to pass through it. That means only phages, that is bacteriophages and smaller molecules could pass, but not the bacterium. So here they didn't allow the contact of N2 bacterium, which was required for the conjugation. Okay, so they and thus and this also was incubated for a certain time period, and then both the sides, like the strains from this side and also the strain from this side, were plated onto the minimal medium. So they found two types of results. First is the minimal. These were the minimal medium which did not have any of the amino acids. Okay, so then they found out that in one plate there is no growth, that is, the organisms couldn't grow because they couldn't produce any of these amino acids, while on some plates they got growth of prototropic colonies which could produce all these amino acids. So, why did this happen? As they didn't allow the contact of two bacteria, the conjugation couldn't take place. So, this transfer was due to a bacteriophage. So, here the agents which transferred the genes coding for the oxytrophic amino acids were transferred into the other bacterium through a bacteriophage. So, hence the process is named as transduction. So, transducing phages. For some bacteriophages, some piece of bacterial chromosome instead of the phage DNA occasionally gets packed in the phage core. These phage particles are called as transduction phages. So it may be not clear here, but as we see the process, we'll get a clear idea what are actually transducing phages. Then the conditions which required to do transaction. So the phages which will perform transaction will need to will need some conditions to follow. The first is the phage degrade the bacterial chromosome. Obviously, the phage should be able to degrade the bacterial chromosome, cut down into its fragments. Then the process of packaging DNA into the phage protein not be specific to the phage DNA. Yes, this is required because if the phage DNA, if the phage protein is specific for the phage DNA only, then then it will not allow the bacterial DNA or the bacterial chromosome to enter into the phage protein. Hence, the transducing phages will not be formed. Third is the bacterial genes transferred by the virus recombined with the chromosome in the recipient cell. Oh, yes, when the bacteriophage infects a new organism, it introduces the bacterial DNA in it, and thus the recombination between the bacterial DNA introduced by the phage and also the bacterial DNA present in the bacteria should recombine each other to form a new recombined recipient cell. Okay. Then we have the actual process of the generalized transduction, which will get, then you will get a better idea of what is actually happening. So, first and foremost, the bacteria infect with the phage. That is, the phage introduces its own DNA, that is the phage DNA, into the donor bacterium. The donor bacterium is the one which is introduced, which is infected by the phage for the first time, from which the genes are transferred. So when the phage infects the donor bacterium, it leads to the lysis of the So not the lysis, it leads to the degradation of the bacterial chromosome here, and then the phage proteins and everything is formed. Then the DNAs, that is the DNA fragments of the phage DNA as well as the bacterial DNA enter into the phage proteins. That so here, as you can see, the normal phage has its own phage DNA, while as the transducing phage has a phage protein, but the DNA inside it is the bacterial DNA. It's not the phage DNA. So such phages are called as the transducing phages. As then as afterwards, when the lysis of the cell takes place, these transducing phages that have the bacterial DNA of the donor bacterium are infected to the new cell, that is the recipient cell. So these transducing phages infect the new cell. So when these transducing phages infect, they introduce the bacterial DNA of the donor bacterium instead of the phage DNA. So when this DNA gets enters the recipient cell, it gets recombined instead of lysis and producing the protein phages. It doesn't perform those functions, so it gets recombined with the phage recipient cell. So if the phage transferred bacterial genes to another bacterium, recombination may take place and produce a transducing bacterial cell. So this recombination takes place here, and we get a transductant, that is transduced bacterial cell. So now the donor bacterium's DNA is recombined with the recipient cell, that is, and a transductant is formed. So the transductant which is formed will have a part of the donor bacterium gene and also its own gene. Thus, transduced bacterial cells are formed and genetic recombination is achieved. So these are the references for my topic. First is the genetics, a conceptual and approach, fourth edition by Benjamin Napiers, and second is the I Genetics, third edition by Peter J. Russell. Thank you for patiently listening this video. I hope you have understood generalized transduction.